In this video, you're welcome. In this video, where we want to look at the complement system under immunology. Complement system is part of the body's immune system. And the word complement means, to complement means you are enhancing. So we see the complement system playing a big role of enhancing the action of antibodies and the phagocytic cells to fight against the body or invading microorganisms and enhancing immunity for our body. So that's why we call it the complement. It complements or it enhances the action of antibodies and phagocytic cells in fighting against any disease-causing pathogen or any other cause of that might be attacking the body. So this complement system, it is made up of, it is a cascade of events. Sometimes we say it is a cascade. You see a cascade of events whereby we see proteins, complement proteins, and these complement proteins are produced in inactive form. So they need to first be activated. Well, we call them zymogens. They are produced in inactive form. And these complements range from complement 1 up to complement 9. And we are seeing all of them are produced in inactive form, so they need a trigger factor to be activated. And we see that whenever they are activated, they form, for example, when I have the complement, it is broken into a smaller subunit and a large subunit, whereby A is a smaller subunit and B is a larger subunit. With the exception, except complement 2, whereby complement 2, when it is cleaved or when it is activated, complement A is the larger subunit, then B is the smaller. So we see complement 2 an exception from other complements, whereby for it 2 is the larger subunit. And all these smaller subunits are cleaved away, and then the larger, they're the one that continue to activate. That's why we are calling it a cascade of the event. One activated step leads to the other. Failure of the first step to be activated will not activate the other. And this complement system, it is both, we shall see types whereby we shall see classical being adaptive, meaning it is specific, whereby we shall see lectin binding pathway being innate, and also alternative being innate, meaning it is non-specific. So lectin pathway and alternative pathway, they are non-specific, whereas classical pathway is specific. So in other words, we are seeing this complement system has 3 pathways. And actually some books call them 4, whereby they include the lytic pathway, which is from complement 5 up to complement 9. So we are seeing classical pathway, we are seeing lectin binding pathway, and alternative pathway. We want to first and all, we want to see what triggers each pathway. We are going to see that the classical pathway is initiated by antibody antigen complex. It is initiated or triggered by antibody antigen complex. This complex, these antibodies are either IgG or IgM. That whenever there is an invading microorganism, we see trigger of IgG or IgM that initiate the complement system. And whenever this complex is formed, it is going to activate. We have complement 1, and this complement 1 has subunits. It has 2 subunits. It has R subunit and AC subunit. So the immune complex comes and binds to complement 1, 2. we see it, this immune complex coming and activating complement 1, 2. And once one complement 2 is activated, it activates complement 1R, and this complement 1R also activates complement 1S. It is the cascade of event. Then the activated complement 1S is the one that goes and activates complement 4 and complement 2, whereby it will cleave, and when they are cleaved, they form larger subunits, whereby we shall form a C3 convertase. And this C3 convertase shall form complement 4B, then complement 2A, because we said A is the larger subunit. If I can do it aside that if I have complement 4, it is cleaved to complement 4A and complement 4B in the presence of complement 1S, which is activated. But because this is a smaller subunit, it is cleaved away, we incorporate larger subunit. Then the same thing with complement 2. So this one is what we call a C3 convertase. This is C3 convertase. So this is how classical pathway is triggered. That in the presence of an immune complex, antibody-antigen complex, we are seeing a majorly IgG and IgM. We are seeing them activating complement 1, 2. Complement 1, 2 activates complement 1R. Then complement 1R activates complement 1S, which goes to complement 4 and complement 2, and they are cleaved to be activated to form a C3 convertase, which is complement 4B, complement 2. That is classical. Then in lectin binding pathway, we are going to see it is triggered by mannose binding lectin, MBL, mannose binding lectin on the surface of pathogens. So whenever there is a pathogen invading the body, we see mannose binding lectin going on the surface of the microorganism. And when it reaches on the surface of the microorganism, it is going to bind it will bind and activate mannose associated saline protease type 1 and type 2, what we call MASIP1 and MASIP2. So in a complex whereby mannose binding lectin on the surface of pathogens are able to go and activate MASIP1 and MASIP2, which is mannose associated saline protease and mannose associated saline protease type 2. And these are the guys that bind together to come and activate. These are the ones that they come and activate complement 4 and complement 2 to form a C3 convertase. So it is the MBL together with MASIP that comes and activate complement 4 and complement 2 to form complement 4B, complement 2A, which is also going to act as a C3 convertase. So we see even under lectin, a C3 convertase is complement B, complement 4B, complement 2A is also a C3 convertase being activated by the MASIP, what we're calling mannose associated saline proteases, type 1 and type 2. Then we are seeing under in alternative pathway, we are going to see the interplay of guys like factor B, factor D, H, and sometimes I. But the H and I, they are inhibitory. We shall see them participating in regulation. So whenever the alternative pathway is triggered by bacterial endotoxin, or what we call microbial endotoxin. So whenever there is an invading microorganism, these microbial, they produce toxins endotoxins are the one that trigger. And what do they trigger? They trigger factor B. But here, if I have factor B, it is cleaved to factor B, small b, and factor B, small b. And what is triggering it? This reaction is occurring in the presence of factor D. Factor D is the one that is doing it. And after this one being the larger subunit, it is the one that is going to form C3b small b. And this C3 small b is unstable. It is unstable. It must be stabilized. And what stabilizes this? We bring in properdine. You see properdine comes in and stabilizes complement 3b, small b, properdine. The first small b, capital B, small b, cleaves some complement 3 to complement 3b. So here there is complement 3b. It is complement 3b. Even this one, it is complement 3 small b, like that. And this is also our C3 convertase. That after being stabilized, it is going to convert more C3 or cleave more C3. So it is stabilized. So properdine stabilizes. So this one becomes stable. And these pathways differ from initial stages. Other stages is going to be the same. So we have formed all C3 convertases. So we are going to see these convertases coming together. And they come together and they activate complement 3 to complement 3A and complement 3B. All of them. So all the pathways meet at C3 convertase. And this, at a later time, we shall see that this smaller subunit is going to act as an aflatoxin. This one we shall act as an aflatoxin, whereby it will go to mast cells and cause degranulation, leading to production of inflammatory mediators. We shall see that. Then complement 3 being the larger subunit, it is going to come and act as a C5 convertase, whereby it comes and converts complement 5 and activates it to complement 5A and complement 5B. And this complement 5A acts the same way as complement 3 as an aflatoxin, and another thing, they act as the aid in chemotaxis. So now we have formed a C5B, which is going to be an initial stage for formation of MAC, which is the fourth pathway known as lytic pathway. So we are going to see, that when we have complement 5B, complement 5 is going to activate complement 6. Complement 6 activates complement 7. Complement 7 says, I will not also be selfish. It also activates complement 8. And also complement 8 activates, and incorporates complement 9. And all these together, when you have complement 5B, complement 6, 7, 8, and 9, they form what we call MAC, membrane attack complex. They form membrane attack complex, and is this membrane attack complex that goes on the surface of blood vessels of cells. It goes on the surface of cells, and this mark having complement 5b, 6, 7, 8, and 9, it comes and surrounds on the vessel, on the cell wall of the vessel, of the cells. And when it surrounds on the cell wall of cells, it begins creating pores. Starts creating pores, starts perforating this cell, leading to formation of pores on the cell wall. That is the mark. We see the mark doing that. It attaches on the membrane of cell walls and causes pores, starts causing pores, and whenever pores are created, we see exocytosis of intracellular content. So the cell loses its intracellular fluid to the surrounding due to the formation of pores. And whenever the cell is losing its intracellular membrane into the surrounding, this one is what results into what we call cytolysis. Cytolysis means cell lysis. Cells are lysed, or they are broken down by the mark, whereby the mark comes and creates pores, leading to the escape of intracellular content, leading to cell death. That is the end of complement system, whereby we have seen it is divided into 3 pathways: classical, lectin binding pathway, and alternative pathway. And we have seen all of them. They meet at C3 convertase, whereby this one is triggered by antigen-antibody complex, and especially IgG-IgM. This one is triggered by mannose binding lectin, found on the surface, which binds on the receptors on the surface of microbial organisms. Then alternative being triggered by microbial endotoxins. Activating factor B. Factor B being activated to BB and BA in presence of proper D. No, factor D. In presence of factor D. Then stabilized by proper D. Proper D stabilizes. And then still it becomes a C3 convertase. After seeing that, I can see how do we regulate complement system. We are going to see there are guys which regulate, guys like, there are substances which regulate this C3 convertase. Guys like DAF, and thioestelase. Thioestelase, they are the one that regulate. They inhibit C3 convertase. They come and inhibit this C3 convertase from converting C3 to C, to activated C3. That is decay activating factor. It is the one that causes the C1 to lose its enzymatic action. So we can no longer activate complement 4 and complement 2. That is decay activating factor and thioestelase being inhibitors of complement system. And another regulator is that these guys are produced in zymogen, as zymogens, in inactive form. So meaning whenever there is no trigger, the cascade of event does not occur. So zymogens also act as the inhibitors. Then another thing, we can always also target complement B. We are seeing complement B being a key player. So if I can target him, I can stop this pathway. And these inhibitors of complement 3, we see factor H and factor I inhibiting, these ones inhibit action of C3b, which is the larger subunit. And lastly, there is on MAC, there are inhibitors that target MAC. Inhibitors like there is protectin for it it inhibits complement 9 from being incorporated into the mark. That is protectin. And whenever the marker does not have complement 9, it doesn't have ability to cause cytolysis. So we see in our body we have protectin that inhibits incorporation of complement 9. And hence, we see it not doing the work of cytolysis. So the inhibitors, we have seen DAF, decay activating factor, thioesterase, inhibiting C3 convertase. We have seen factor H and factor I, inhibiting the action of complement 3B. And this complement 3, when we are looking at the significance, we are going to see it is a strong opsonine. It is an opsonin. It has a role of opsonization. Then we have seen we can protect addition of complement 9 and complement 8 by protecting, and with that one we can prevent this complement. Lastly is the significance. Why complement system? We should always know why is complement system very important in our body. That is the last thing we can talk about, significance of the complement system. And we have seen that this complement system, it was producing complement 3A and complement 5A. These guys, they were acting as nephrotoxins. How they go on the surface of mast cells and trigger mast cells to release histamine, that is inflammatory mediator, and leukotrienes. And these histamine produced by the mast cells, they are the ones that are going to enhance inflammation, they're going to trigger inflammation, and we see leukotrienes doing the work of chemotaxis. So far we have 2 points here. Complement system is responsible for triggering inflammation via an aflatoxin and also triggering chemotaxis, whereby the cells are able to move towards the enemy in a unidirectional movement. Then another significance, this is number 1, number 2, number 3, we saw complement 3B acting as an opso, doing the work of opsonization, whereby opsonization is like tagging, mark, or you mark. It marks those cells which are affected or the microorganisms, so that when the CD4s and the phagocytic cells are coming to kill, they kill only affected cells. So meaning opsonization enhance phagocytosis. Then we have seen number 4 being the mark, whereby the membrane attack complex was doing the role of cytolysis, cell death, killing cells. That is the major, these are the major significances of the complement system. Even number 5 is that these guys regulate the immune system. How? By clearing or removing immune complexes. That whenever there is immune complex in the body, they can be cleared by the complement system. And now you can imagine, if I'm deficient of any factor of the complement system, you can imagine what will happen. We see deficiency of the complement system. We see people suffering from autoimmune diseases, and these autoimmune diseases include systemic lupus erythematosus. Whenever you are deficient of complement B, or the complement system is not working, you suffer from an autoimmune disease known as systemic lupus erythematosus. And another thing we have seen, they are playing a big role in preventing microorganisms from entering in the body. So whenever you are deficient of the complement system, we see you suffering from microbial diseases like Neisseria gonorrhoeae. So you are susceptible to bacterial infection. We see infection is coming into play because there is no enemy to fight against the infections. Thank you so much for listening. Always remember to subscribe, share your comments, and like the video so that we can continue to grow. Thank you so much.